for this request.
Welcome to today's Lazarev seminar. It is my pleasure to introduce Daniel Lazarev, who is currently a PhD candidate in mathematics at MIT. His research involves the development and application of marking information theoretic methods to infer structure, interaction, and dynamics of biological and physical systems. Today, he's going to be talking about entropy-extremizing representation with applications to biology.
All right. Thank you very much. Very excited to be here. Thank you, Sreshta and Nate, for the organizers, for the invite. Thank you, Jonathan. The whole department, very happy to be here. So I actually defended my thesis a month ago, so it was actually good. I could give you some snippets from what I used in the defense, not the entire defense, hopefully some interesting things that are relevant to the seminar. And also want to tie it in with some kind of larger ways that, thinking about the field and thinking about where things are moving on a personal level and also in terms of math and biology in general. So my own background, so I studied math and physics and pre-med in undergrad, and I started med school in 2018, and I started grad school in math in 2021, and the staggered lines are because I actually fit in some of the last requirements of my school during my summer and winter breaks. So I had a little bit of an non-traditional background, but always fascinated by these connections between mathematics and biology. And in particular, I wanted my training to reflect the ability of kind of this pipeline going between theory and applications. And I think application-driven research is the most exciting and actually drives math. That's the most meaningful. And also, as I kind of went about this route, I actually learned that some of my personal heroes, both on a personal level and as a scientist, actually had backgrounds in both math and biology, or math and medicine. One of them is Maimonides. So he was famous for his Jewish writings, but also for his medical writings. He's also an astronomer, a mathematician. More recently, so he's actually his relief in the House of Representatives. He had an impact on even in America in other ways. But other mathematicians that we know, like Cardano, was actually also a physician known for the quartic equation, quintic equations. He's also of course, a mathematician, but also a physician. And you see that the kind of the scientists of old cared about these kind of applications in the deep level. Historically, it's actually interesting. A lot of them were physicians for a technical reason, which is because in the old university system, the first few hundred years since the university started in about the 10th or 11th century, you could only obtain a doctorate in theology, law, or medicine. So if you're a scientist, you automatically got your doctorate in medicine so that you could teach, and math was not really an established field at that point. So there's actually also a technical reason that a lot of them got medical training. But he actually made most of his money from gambling, so it informed his developments in statistics and probability. Galileo actually left medical school to focus on physics and math. He's known to be fond of throwing things off buildings. And lastly, as an example of Bernoulli, also a famous example, his dissertation was actually in the medical physics of respiration. He's one of the rare cases where we have a before and after picture, before grad school and after grad school. So I kind of, his education did a number on him. So kind of comparatively, maybe, I'm okay. But this essay by Wang in October 1948 was really transformed the way I think about it, in a way kind of I place my own research goals and ambitions. So he roughly divided scientific development into different eras, both conceptually and also historically. So the first was organized simplicity, so the 18th to 19th centuries. And this it's simple because it involves only a small number of variables, and it's organized because they're non-randomly interacting. So that was used to model planetary motion, rocket science, kinematics, mechanics. All those developments happened because of these kind of models and mathematical developments, and involved the development of calculus, differential equations. Further down the line, we had the complete opposites. You had disorganized complexity, so billions of variables, and they're randomly interacting. So the fact that they're randomly interacting allows you to make certain simplifying assumptions and certain developments in thermodynamics, theory of gases, statistical mechanics, and statistics and probability kind of fall in this category. The next frontier, as Warren Weaver put it, was actually in organized complexity. And that was what his essay was really about what do we do looking forward, and how do we deal with this next, the most difficult area. And this involves many variables, but they're non-randomly interacting. So this is actually the case in biology and medicine, in fluid dynamics, turbulence. That's what makes these problems so hard. I mean, a lot of famous mathematical problems in fluid dynamics and turbulence, non-equilibrium dynamics. And what he actually said at the time was that what's going to drive this progress is progress in computing. And he was largely right? So high performance computing, AI today. But what I'd like to argue is there's actually other areas of math, new math that needs to be developed to properly tackle this. And I think this has to do with finding optimal representations of these systems. And we're going to talk more about what I mean when I say that. And also this entropy theory kind of perspective. So really you're kind of looking at three different axes today, whereas before you consider theory and application, now there's this interaction with computation as well. And between the three of them, you want to put together something that allows you to solve these complicated problems in biology. So really, maybe I don't have to convince this group, but biology to math today and going forward is going to be probably what physics was to math in the last few hundred years in terms of driving progress and being a source of interesting problems and data. So this is a paper or a book by Bernoulli, and he's famous for his Bernoulli's principle or other works in hydrodynamics. And this is really energy conservation, right? Another way to frame this is in terms of energy conservation. So you have a situation where a simple variational principle can actually drive a lot of progress in terms of solving problems. And in other contexts, similar kind of things hold. So the Navier-Stokes equation for fluid motion and turbulence is a reflection of momentum conservation. So my first foray into this kind of work was during medical school. I worked with a mathematician who the driving problem was modeling turbulent blood flow through the heart. And it turned out that the solutions, when you actually try to forget about solving it on a mathematical level, but even just simulate it, really depended on your grid size. So you're seeing the existence of non-unique solutions. And that's a big problem if you're actually trying to solve a problem like, seeing which heart valves work and how to solve certain diseases that are caused by turbulent blood flow, which is what this picture is showing. And what we did in that work was use entropy maximization as a way of singling out the physical solution. So the equations themselves are underspecified. You include this extra condition, you're actually able to this criterion helps you actually find the real physical solution, and with all the downstream applications. And this kind of got me thinking about really what entropy is. Why is it? I started reading about entropy in different contexts, about the literature it's used in maximum entropy in statistics and thermodynamics, but also in other contexts throughout math. And it seems like it's almost this magical thing. People kind of introduce some notion of entropy, and it drives all kinds of progress. So there's examples that we see here. There's examples we'll discuss later. But it really seems like this mysterious thing. So first of all, I'm gonna, certain parts I'm gonna run through a little faster because I want to get to the things that are actually interesting. But people know about Shannon entropy. Some intuition about it. It measures the number of states that are possible with some information. So it's actually the log of that number. So if you have a fair point, that's log of 2. So you have 2 equal possibilities. If it's an unfair point, probability of 1, you get an entropy of 0. So you only have one possible state. I'm going to just run through this a little bit, but really it gives you entropy. It gives you a way of measuring the effective number of possibilities you have in your system, and you could view it as a measure of uniformity, randomness, uncertainty. All these different words have been used to describe it in some sort of fuzzy way, but hopefully it could be some more concrete way we could talk about it. So Shannon entropy, you have both discrete and continuous versions of it that appeared even in Shannon's original paper. I could get this out of the way a little bit. You can actually describe it more generally in the language of measure theory that actually encapsulates both these notions. And then there are other entropies. There's Renyi entropy. So here I actually wrote it. It's usually not written this way, but this I wrote in terms of the measure theoretic description. And this, in the limit of p going to 1, reduces to Shannon, but it actually gives you a wider family of entropies with different properties. Talos entropy, which came from the physics community, is very similar to Rényi, except that you don't have a log term, but it also, in the limit of P going to 1, also equals Shannon entropy. And then you go throughout math, you see kind of more abstract notions. So there's topological entropy, there's measured theoretic entropy, Kolmogorov-Sinai, relations between them. There's Furstenberg entropy in group theory. Perelman entropy is interesting. It was actually introduced about 20 or 24 years ago in the context of solving the Poincare conjecture. So Perelman showed that this quantity obeys some sort of monotonicity property, and that allowed him to prove the conjecture. And that's a pattern you see. You see people introducing these entropies, and they're, somehow do some sort of magical thing, and no one has a good explanation. So when I really learned that was my first year of grad school. So Kolding and Minicozzi are two mathematicians in the math department at MIT, and I took Minicozzi's and Kolding's geometry classes, and they introduced this notion of entropy in the context of mean curvature flow and studying minimal surfaces. And I asked them, they got fantastic results through this, also monotonicity and being able to characterize these minimal surfaces. And I asked them, really, how do you come about this? They knew Perelman personally, they know the field. How do you come about these definitions of entropy? And they basically said it's almost a guess and check, right? There's no organized theory to kind of get at how you define a certain entropy for a given context. Yeah.
I have never seen so many definitions of
entropy
in one part.
Yeah.
Why are they all called entropy? What is the basic common?
That's exactly exactly. That's what I'm going to try to get at. So that kind of, it was the actual driving question behind kind of this line of work and further down the line as well. Oh, sorry. Sorry about that.
Yeah.
Sorry.
And maybe
Okay. Is there a way to maybe hide it? Yeah. All right. All right. There you go. So they're obviously more than introduce anything. Really, the point of these slides was just to show you that there's a bunch of them. And something, what's the point of calling it entropy? So one would argue that maybe you're just calling it that because no one knows what it means. So you're always you're okay. If you call something a quantity you don't know that maybe has a logarithm in it, entropy, you're, in the words of our Neumann, you're going to win your arguments half the time. But the driving questions is there a unifying framework that encapsulates the known entropies, but also allowed for a principal formulation of new ones in a new context of application? And fundamentally, what does entropy mean across these contexts and why it's useful? I'm going to run through this. This is kind of more some mathematical developments in this space, but I wanted to drive the message home more than the actual content in this part of the talk. So you can actually formulate entropy in the context of what I call your structure spaces. So this is a way of encapsulating this or providing a context to describe these different theories or applications. So you have some sort of underlying set, you have some sort of experiment space associated with it, which could be a product of the spaces or or a power set. And you look at the structure as a map or a subset of maps from this experiment space to some sort of target space that somehow gives you kind of the structure you're interested in. So a few examples of that. So metric spaces fall in that paradigm. So your experiment space is going to be the product, your target spaces are, and your structures are these metrics that satisfy these properties. So the same thing with groups, where your structure now is the group operation. Same thing with topological spaces, where now you're looking at this subset operation, the open sets. Same thing for measure spaces. And the point is that given this context, you can now say, okay, what is entropy in the context of a general structure space? And now if I kind of try to crystallize it, a given structure, what comes out? And so the thing that in terms of like seeing patterns, I think the defining feature of entropy is that you have some sort of ordering relation or pre-order. And entropy is just a coordinate on that. And it gives you a way of actually, the monotonicity property of entropy is really what defines it. And everything else is kind of comes with once you actually crystallize the given structure. Yeah, meaning so you have some sort of ordering relation that gives you a sense of something more structured or less structured. And entropy gives you a coordinate along R that actually allows you to quantify that. And turns out you get some sort of results. I'm not going to go over this, but in terms of generalized families of entropies, you get a universal entropy theorem that gives you the existence of such an entropy and the fact that all possible entropies are some order preserving map away from each other. And then the other one is an entropy rigidity theorem, which once you introduce the maps between the different structures, you could actually get to a more limited family of entropies, and you could actually derive some of these entropies we looked at. So the constructional, like I said, allows you to derive these given the context, and opens the door to the definition of new entropies tailored for new problems. So depending on how much time we have, so I'm gonna maybe skip the next section, but it shows you basically in the context of Stokes theorem, an example of that. But let's talk about optimal representations. I'll maybe skip some part to allow us to get to the things I want to talk about most, and that more application side. So when we talk about optimal representations, what do we mean? So this is the model of the solar system. If you put Earth to the center, it's not, I mean, we say it's wrong, but it's not wrong per se, it's just more complicated, right? So if you had to measure our months or measure things in the solar system based on this model, we'd have a really hard time because the calculations would be incredibly complicated. What the heliocentric model affords us is it's simpler and it's easier to work with, and it actually kind of gets at the underlying structure of the solar system, right? And a lot of the calculations become that much more simple. So the optimal representation in this case, what makes this kind of more real is that it's in some ways encapsulating the underlying information in a more straightforward way. Same thing, you know, if you want to play basketball and you're looking through a fishbowl, you're going to have a hard time actually measuring out your trajectories, right? So you might have to get some corrective lenses. If you actually look through kind of the way we see, you might also see some features that were hidden before, like the fact that this hoop is not really like a real basketball hoop. It's actually 3 different hoops. And that might confuse you as well. And in different contexts in math, we actually use optimal representations, and, you know, to make problems tractable or even solvable, right? So in the context of, you know, exercise you do in undergrad, you know, you want to solve the Gaussian integral. You put it in radial coordinates, which uses the spherical symmetry of the problem, and you actually are able to solve it exactly. So, you know, this quote by Grothendieck, you know, you should never try to prove anything that's not almost obvious. So the idea is once you, even in theorem proving, once you put something in the appropriate representation, things become easy. Things become straightforward. And, you know, that's a theme throughout math, right? Transforming a hard problem into a space where it's easier through a Fourier transform, through whatever it is you're using. And then, you know, once you solve it in the optimal representation, you kind of ship it back. We're going to see how that plays out in applications to biology immediately after this. I'm going to skip this part just for reasons of time. And so this project, this work is basically trying to find lane representations given genetic data. More specifically, this is using genome-wide association studies. So summary statistics from things like the UK Biobank or FinGen, whoever is familiar. And the idea is you have these large matrices, the summary statistics relating a large group of genetic variants, so SNPs, single nucleotide polymorphisms, and some sort of huge set of traits. And we want to learn, right, given this big matrix, you want to learn some sort of intermediate representation with latent factors, and in some ways encapsulate the underlying biological mechanisms or biological insights that we're trying to get from the data in a way that is purely driven by the data and doesn't need the user to specify any, you know, application-specific hypotheses, right? So the current paradigm in biology is you know, the traditional kind of scientific method, right? You have some sort of hypothesis and you check it, but that's very hard when you have, you know, tens of thousands of biological traits. And the hypotheses have to become more and more complex to actually be able to get at the underlying mechanisms, which themselves are not really simple. So the basic problem is one of, as a factorization problem, in essence. So you have matrix beta of summary statistics, you have M SNPs, these genetic variants, and you have T traits. And typically the number of latent factors is going to be less than both of them. And you're trying to decompose it in such a way that these weights multiply back into the original summary statistics matrix. Really, it turns out to be useful to separate out the scaling. So you're looking at, we'll get to that. So yeah, so I'll explain why maybe in a little bit, but basically if you separate out the scaling, these become, these WXL and WYL matrices are essentially picking a rotation, right? To actually a rotation and a truncation in one step that transports you to this latent space that you'll then add the scaling to make it work. So a possible solution is just a truncated SVD, which is very natural. So you start with a full SVD, you truncate it to some L, and then you identify the weights or these rotation matrices as the U and V. However, right, these solutions are unique up to some sort of orthogonal matrix, right? So you could always multiply inside by an orthogonal matrix and it's transposed, and you get another possible solution, right? Namely, your WXL could be UA, your WYL could be your AV, right? VA, and then whatever's left over is going to be your scaling, right? And the question is, can we find some sort of optimal basis matrix? So what's the optimal rotation that's going to give us the most bang for our buck? Is going to be able to extract all the information that's really hidden in our summer statistics matrix. So this is just a commutative diagram to make sense of the different things we're talking about. There's a lot of notation, and just to make sense of what we're looking at. So whoever's not familiar with commutative diagrams, it should be the case that no matter which path you traverse, it should be equal in the end. So one way is B, which is the matrix taking me from the input layer into the output layer, should equal this inner loop, which is the SVD solution. So U times S times V transpose. So that should be equal. And we're saying more generally, if we have a different lane layer, we should be able to go the WXL route, the outer loop, through some other scaling matrix, and back to WYL. And based on how you can actually see that WXL has to equal U times A, and WYL has to equal V times A, and make this all work. Of course, figure out that sigma should be A. But that's kind of just to make sense of the space we're working in, where they're, what they're embedded in, and so on. So in this context, ICA, independent component analysis, is an important tool to implement what we're trying to do. So just to some background about ICA. So first off, in general, entropy is maximized for a Gaussian distribution on R. So if you look on the real line and your only constraint is a finite variance, the maximum entropy distribution is a Gaussian. And when you mix non-Gaussian signals, they become more Gaussian. So the classic problem where this ICA is introduced usually is the cocktail party problem. So you have a recording machine, you come to a cocktail party and people are having conversations. And the more people you have, the more your recording is going to sound like white noise. So you want to be able to separate out the individual conversations from your recording. And the idea is you essentially do the opposite of the central limit theorem. So there's this nice paper from 20 years ago where they finally proved a conjecture by Shannon that entropy increases at every step as you are in this kind of limit towards the Gaussian and the central limit theorem. So the idea is essentially if it increases at every step, if I minimize entropy, I'm able to go back to that original frame of the conversation being separate. So you kind of, the goal of ICA is essentially to do, one way to frame it, people usually don't talk about it this way. I'll show you in a second how they usually kind of talk about it, but essentially what you're trying to do is reverse the central limit theorem. And that allows you to, this is a common picture from scikit-learn that implements this. You're actually able to reconstruct the original signals this way up to a sign permutation. So this is essentially one way to phrase it. I phrased it actually in the context that we applied it in. So what we were trying to do to find this optimal representation, we're trying to find a basis that minimizes entropy. So then we realized that ICA, essentially, if you use it in the right way, you could actually, instead of having to write the code from scratch, you actually could apply ICA in a specific way that does exactly that. So it essentially, let's see. This is actually how we implemented it. So this is not a particular, this is not ICA in general. This is, we actually found an A that minimizes this. I'll show this in a second, but this concatenation of the U and V matrices so that your basis is simultaneously informed with the input and output maps. But the idea is that you're minimizing entropy and just in order to extract the most information. Because when signals are mixed, you lose information. Okay? Usually ICA is discussed in terms of neg entropy, and neg entropy, all that is the difference between the maximum entropy, which is the Gaussian, and the entropy of your data. So maximizing neg entropy is the same as minimizing entropy because this entropy, the Gaussian, is fixed. In practice, ICA models rarely actually do an entropy minimization for computational reasons. So if you actually had to minimize entropy directly, you'd be estimating a PDF at every step, which is very expensive. So instead, what they do is they look at certain proxies. So an early proxy that was used was kurtosis. The problem with kurtosis is that it's sensitive to outliers. So then, there's a lot of work that was done to find other proxies that work well. If people are familiar, the main implementation of ICA now, the most popular one is fast ICA, developed about 25 years ago. And what that did is they found optimal, what I call contrast functions that they found actually using a maximum entropy approach. So you got entropy here figuring in every step. So this is the pipeline in our method. So you start with an SVD of your beta matrix. You get some sort of SVD solution. You take the input and output rotation maps, concatenate them, and then you apply ICA to this concatenated map to find the rotation matrix that minimizes entropy. Once you have that, you can now construct your optimal solutions that are entropy minimizing. Yeah.
So it's kind of reminding me of, say, a backward heat equation, which is known to be not a well-posed problem. So maybe you could just say a little bit about, here you're kind of presenting maybe a best case scenario where this sort of works. Maybe you can say a little bit about some of the constraints and when it's not working. So.
Yeah. So if your underlying signal is Gaussian, it's not going to work because basically, and it's actually not a bad thing. It actually allows you to sometimes do something helpful, which is remove noise, which is usually in this, Gaussian. But if your underlying signal is Gaussian, you're basically, so ICA, as it's implemented, it initiates the algorithm with a random matrix. And because of the spherical symmetry of Gaussians, there's no particular rotation that is favored over any other. So you essentially end up with a solution being a random rotation. I wasn't going to talk about this, but this actually could be used in a good way. So one part of this work that I wasn't going to talk about was actually estimating the optimal latent dimensionality. And what you could do is you could run the ICA step many times and then correlate the different A matrices, the basic matrices. And the real non-Gaussian signals are going to be Stein permutations of each other, and the noisy part, which is Gaussian, is just going to be random correlation on average 0. So you could use that in a positive way. But if your underlying signal is Gaussian, then this is not going to work.
And, you're kind of saying that in some situations it's not going to give you a unique solution, but it seems like that's more general. Like maybe you don't expect uniqueness in a whole range of types of problems. So
you're saying,
could you say a bit about, even just at the level of the linear system, like, is the condition number blowing up? How is that looking just in terms of, like, since you're solving a problem that's not,
it's not
quite well posed, at least at a continuum level, that should show up in
when it doesn't work, what it doesn't do is it doesn't confabulate. So in the case of a linkage disequilibrium LD, we check different solutions. So like SVD, for example, will show you solutions that seem to have significant weights when the ground truth is not. With this, which is based on ICA, you do not get that. So if it doesn't converge, it just doesn't converge. So you just don't get a solution, or you get a solution that gives you insignificant weights. So yes, there are, you have situations where there's no solution and it'll give you something random, but it's not going to confabulate in a way that actually maybe drives the science, right, the application, RI. Does that make sense? So yes, situations where it just doesn't converge. Yeah, I agree. Sometimes it converges for certain components, but not others. So you could have situations where certain lane components are good and they converge to a solution, but not all of them. So the ones that don't, you just don't use, or you just can't use. But the ones that do could actually give you some good insights. So there's some questions here fundamentally that just need more work.
But if I'm understanding correctly, you're saying when it is converging to something, there's some robustness. So even if you were to start with some different initial conditions, the components that are converging.
Yeah.
And so, I don't
have mathematical guarantees of that. Right. On the theoretical, I don't have mathematical guarantees of that, what you're saying. But in practice, having done this many times and checking it, once you get a solution, and we've done a lot of empirical work to check that. Once you get a solution, it's reproducible in a very real way. Even I'm going to have some results here, even across different cohorts and different studies. And they're all signed permutations of each other, which you can correct for. Yeah. Yeah. So this is these matrices. Yeah. So the solutions you get here are very sparse usually.
Yeah.
Right. And that's due to this entropy minimizing frame that actually does that by design.
But that's the information.
Yeah. And I'll show examples of like real biological.
I'm just wondering, does sigma now have anything in it? What?
And so sigma is scaling, but it's not scaling along a kind of that. It's not a diagonal matrix. So a lot of people usually ask, in what way, this, so SVD maximizes variance, right? That's well known. But it maximizes variance along the frame that you map to, like in your latent layer, right? In this frame, you're maximizing the variance, and it has to be along the axis. Here, you're maximizing a different notion of variance, which is that due to your mappings themselves, not due to your target. And that actually ends up being more useful in biological applications.
But at the end, you can throw away sigma once you get the W.
So we use it to weight these. So these are fundamentally orthogonal matrices. So you use them to weight them to actually have scores to your network. Yeah. So you do multiply them back in. Sometimes you don't use them either, but yeah. I'm going to talk about this later. There's actually related work to basically signify or to compute the significance of the learned weights. So I'm just going to. And that's actually a different part, but really what we want to get to is this network representation. So now you use these weights now to build your network. This network ends up being much more sparse, and then we can run all kinds of downstream analyses, and it ends up being more biologically interpreted. And you could actually train LLMs to work on to actually analyze these and give you insights in an automated fashion. I mean, we're building models. The one I'm going to show you has only 180 factors, but we're also building with 5,000. So now you can have LLMs kind of run through it and give you biological insights. We train also in the literature to kind of do this in a more principled way. I'm going to run through. So this is some simulations. I could recreate or rederive the ground truth weights up to assigned permutation. I'm going to skip over this because I want to get to some of the applications. So this was I was talking about earlier. So this notion of variance components has to do with, it's a metric that uses these the weights I was talking about earlier. So the G, the WXL times becomes the sigma. And you actually capture more of the signal, more of this variance component of the mapping with guide with this method than you do with, let's say, TSVD. We actually compared also to other methods just out of the spot. So your top one component captures more than SVD, even though in a certain sense, right? This is where people usually ask me about kind of the variance aspect of SVD, but it comes with this distinction about what variance we're talking about. And same thing on the trait side. So this is on the variance side. This is on the trait side. So you have, vice versa. That was on the trait side. This is on the variance side. So you get more sparsity. So you have more less SNPs loading onto a given trait, latent factor. And kind of zooming in a little, if you apply this to particular traits of interest, you actually get these weight scores in your network. So guide is our method, genetic unmixing by independent decomposition. TSVD is the one based on truncated SVD. Factor V is a comparable factor model that uses a VAE to also learn these independent factors. And this is kind of, this is kind of the best comparator for this kind of work. So zooming in on, so these are a bunch of traits that are in the UK Biobank in the data set that we used here. Zooming in on 3 of them just as a kind of case study. So on Alzheimer's, you see that there's one latent factor, this latent 72, which has drives a lot of the signal for Alzheimer's. For high cholesterol, you have this latent 10. And for peripheral vascular disease, it seems like they're all comparable, right? So you're seeing for peripheral vascular disease, you're not really getting much signal there that you would get with just a TSVD, which maybe tells you there's either underlying gaps in signal or that the method didn't really give you anything useful for that lane factor. The lane factors for those traits. What's interesting is that, so the guide lane factors, not only are they more interpretable, but they're also self-consistent. So we're going to see how we're going to identify these different lane factors, but they carry the same function across the different traits. So lane 72, we're going to see in a second, is this ApoE lane factor. So loads very strongly on the ApoE gene, and then it's very specific for the Alzheimer's disease trait. The lane 10 is a general cholesterol metabolism pathway kind of lane factor. So it's less important. It still appears as the second most important lane factor for Alzheimer's disease. Not quite as important as this ApoE lane factor, but it's a kind of secondary role. But it's the most important one for high cholesterol trait or for statin drugs, right? So things that are related to cholesterol. Fundamentally, lane 10 is always the top one, followed by lane 72, because ApoE does have a role in cholesterol metabolism. When you look at other models, they're not self-consistent in the same way. So for TSVD, it's lane 71 and 64 here. It's 21 and 19 when we look at high cholesterol. And, you know, here actually there's a little bit of matching. So lane 21 actually did have some features that lane 10 has. So sometimes you do get a situation where the TSVD rotation for some lane factors is almost the most optimal one, almost kind of as a fluke. That's not a common result, but you can see that, which is why I also chose this example. But you get completely different, in general, you get completely different lane factors for these other models, but you get a self-consistent picture with guide. So this is we host this model online if anyone wants to check it out. So if you look at the drop down for Alzheimer's disease, you get the breakdown for the lane factor, the most, so the highest kind of the biggest proportion of this lane factor is dominated by lane 72 for this trait. And lane 72 on the trait side, which is this top plot on the trait side, lane 72 loads on to mostly Alzheimer's disease, a little bit on high cholesterol, and a tiny bit on statin. This lane 10 that I told you is this cholesterol metabolism trait. It's the traits that characterize or that it interacts with the most are high cholesterol, statin, which is a cholesterol drug, and simvastatin, which is an example of a statin.
Coffee intake.
Where is that? Oh, so coffee intake is a trait that they track. So some of the traits that have to do with the blood pressure actually have some coffee intake, and also for cardiovascular health. We're going to see some examples of that later on. So the way you could kind of see the pipeline of using this is you build a model, you learn the lane factors, you see you have a certain lane factor, a certain trait of interest, you look at the lane factors and load them, and then you characterize them based on the other loads. So in our case, this is Alzheimer's disease, we trace it back to this lane 72 and lane 10. Now we look at lane 72 and lane 10 in terms of both their loadings on other traits and our loadings on other genetic variants to see how we could characterize it. So that's kind of what we did here. And then on the variant side, this is the plot you get. So the locus that most highly loads on the lane 72 is the one that is 19q13.32, which is the one that has ApoE. You have some other things that are much more minor. The lane 10 is actually more spread out, more evenly spread out, and all of these are essentially different genes in the cholesterol metabolism pathways. You have LDLR here, you have also the same locus with we and ApoC, you have ApoA over here, LPA, so PCSK9. So you have genes that you kind of make sense to be part of this and cholesterol metabolism lane factor. If you look at another set of traits, so we looked at forced expiratory velocity in 1 second. So measure of lung health. So this is a test that's done in the clinic where you kind of blow into a tube and they measure how much air is being blown out in 1 second. It's usually done for smokers to measure their lung health. So in this case, it's a much more complicated multifactorial kind of trait. So we looked at seeing can the lane factors separate out the different components that contribute to it. And I'm not going to go into all the details because it does get more complicated, but you have these lane factors that are more specific for FEV1. And you look at it's hard to say just from this how to characterize them, but you look at these, let's say latent 7 on the variant side, and you actually see that, okay, on the variant side, it's all of these immunological variants that make, so you can characterize this as an immunological latent factor that kind of drives this diseases that have to do with the lungs. Other things that are more interesting here, let's say like grip strength. So latent 65, we found that we characterize it more as a cardiovascular health latent factor. So on the variant side, you'll see things like Titan, right, or things like that have to do more with cardiac health. And then you have another latent factor that prioritizes sitting height or trunk size. So basically, you could actually kind of fool the test because you happen to have a large trunk and you're just tall. So you just blow out more air because you're a larger person or you're a smaller person. So that also factors into the separate latent factor. And another thing we tried to do here is see the effects of removing certain data and how that influences the model. So one thing we're interested in here is we identified a factor that had to do with smoking behavior. And then we took out the RNA5, the nicotinic receptors that are implicated in this kind of addictive behavior. So we took out that part of the summary statistics in the data and ran the model again, and then that latent factor that has to do with smoking behavior disappeared. So you could actually, your model it really builds this entropy minimizing representation that given whatever data you feed into it. So it does as well as the amount of data. So it kind of maybe favors an approach that puts in as much data as possible. Then you might run into questions of convergence, right? But when it does converge, you get kind of this complete self-consistent picture that really reveals these biological insights in an automated fashion. This is a Manhattan plot of the features for the top this is a Manhattan plot of all the variants in the study. And we color-coded the variants that are implicated with the top 3 latent factors for FEV1. And we found that they're 24.5, 25 times more likely to implicate the significant loci. So if you have this crude measure of enrichment, where you basically take a ratio above the threshold, the significance threshold of 5 times 10 to the negative 8 over here, and you divide it by the total number of variants, you get this ratio, so of 25 times. So the ones that are implicated by our model, so among all variants implicated, among all the colored variants, the ones that are over the threshold versus all the points in the plot is this ratio of 25. So you kind of get this enrichment if you were to use the variants that are implicated by the latent representation. In terms of quick reproducibility point, so we looked at 2 different models, one built using the UK Biobank and one built using FinGen. So this is a data set that was obtained from the UKB FinGen MVP data set. So they actually made sure to harmonize the results for the same genetic variants and on the same set of 310 traits. So they actually allow a comparison. So we built different models, different number of latent factors, and we looked at how they compare after you adjust for certain arbitrary differences, like a permutation or some sort of rotation in the actual expression of the data. And you found that to a large extent, and there's some metrics here also to show this. So this is on the variant side, this is on the trait side. You found that they're largely reproducible for a lot of the latent factors. And this is going back to the GUI. We actually have also an LLM characterization tab where we did what I said earlier, kind of feed an LLM with all this information and allows you to actually get a description. So you get the variance component of the top phenotypes for that given latent factor. You get the genes that correspond to the variants that load on it the most. And you get sometimes some kind of key gene function annotations that allow you to characterize, the key drivers behind the mechanisms that you're looking at. And let's see if I had this here. So I do want to allow time for questions or discussions. I know I was going to discuss this W value framework, which is what I mentioned earlier about the estimating significance of the learned weights. But if there are questions, we can maybe take them now and see if we have time. Yeah. 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 Yeah.
are those each individual, number of those, I see the other, but are the number of those completely determined by data, or there is some prior,
The number of the,
like, the number of those colored lists from forced expiratory to.
so these are, yeah, so we included the top 3 traits for every latent factor. That's what we did here. So this is just the top 3 for every single one. Yeah, top 3 traits for every single latent factor, and these are the latent factors that are kind of have the highest score in terms of their loadings onto FEV1.
Another really good question. So your representation is not sparse, so it minimizes. Okay.
I mean, it is sparse in the sense of the weights, these WXL, WL, YL matrices. So those are by design as sparse as they could be, where you minimize entropy across kind of both maps simultaneously. So if I apply it to one side, I could make it more sparse, but then I'm not accounting for the information on the trait side. So like in a harmonized way, that's the most sparse it could be,
So it
even
is.
in principle.
So that's kind of each latent variable has a minimum.
Yeah, so that's this part, yes.
Yeah.
Yeah. And there's a whole other part about kind of estimating the latent dimensionality. That's I talked a little bit about one way we do it. There's other things that we've also introduced as part of this framework. But there's ways of doing that kind of, I mean, right now I hit it in the initial, truncation step, but there's actually a more principled way of doing it that is also informed by the data structure.
So here the failed colors are.
They're like, yeah, they're more or like smaller. Yeah, this is just a mental. Was
Did
that?
you cleave it to 0?
No, this is just based on after we apply this rotation. So if you have a few minutes, I'll maybe run through this framework, which was developed as part of Guide. As kind of this actually, the framework was initially, I thought of it as an answer to a reviewer actually, who said, how do you quantify the statistical significance of the weights that you're learning? And then I realized it actually is applicable more broadly in machine learning as a way of quantifying network weight significance. So the idea is that, so this is kind of a representation of maybe a multi-layer network. So something you might be seeing with neural networks or other architectures. And the idea is you could normalize the weights. And once you do, they lie on a sphere. Once they lie on a sphere, you could, assuming that they're uniformly distributed, you could look at the marginal distribution of a single coordinate of that, of a kind of this vector of weights, and compare the weight you actually see with what you'd get under this kind of null model of all possible models that exist, right? All possible models, meaning the ones that they all have to lie on the same sphere. So this is just kind of high level, but I'm gonna kind of maybe explain that more closely. So if you consider 2 layers adjacent to each other, just to simplify the notation. So you have layer A or math-cal A and B. And you consider these weights that you've normalized. And this is a particular A from little a as a member of A and little b as a member of B. And this is a vector of length, the whole size of your output layer. So one way to see it is that this is a point. Also, so essentially this starting from some particular point in your input layer to all the points in your output layer, that vector is once you normalize, is a point on this B-1 sphere. And the set of all weights is this collection of points on that same sphere. So you consider all possible models, right? They're different colors here. So these are all different models of in the same network. They're all essentially collections of points on the sphere. And the idea is, what's the distribution, assuming that we have a uniform distribution on the sphere? So if it's uniform on the sphere, the particular weight is distributed according to a beta distribution. And that's essentially the key part. I'm gonna maybe skip this part. So actually in the context of neural networks, you actually want to do this not in a one-sided way, but in a two-sided way. So you're actually looking at not as a model being a collection of points, but being a point on a larger sphere that's based on both the input and output layers. It actually ends up being a little more useful. But the long and short of it is that the squared weights are distributed according to this beta with these exact parameters. And this what we call the W value is the probability of seeing this, the way that you're actually measuring, or a more extreme value than it. And this I'm going to skip through this, some work in the context of some of the neural network applications. But the idea is you could actually, by this framework, start building QQ plots with your network weights, which is something that you can't traditionally do with in machine learning. And you can actually judge the significance of your weights much in the same way as we do with traditional QQ plots. And this is kind of this is a more recent one. So in this plot, sorry, it's a little small. Should have made it bigger. But what we try to do is two different things. So we start with a weight at initialization before you're learning, and then after training. And you look at it, this model, this is based on a simulation where we had two features. Two of the columns were actually the features, everything else was kind of noise. And so this is the distribution of all the data. This is the distribution before and after training. This is the distribution of the actual real features. So you see how they start deviating and having more significant weights. These are the corresponding significance values, these W values that we talked about. So you see how they actually are able to quantify these things. And the idea is there are two ways you could find significance here. One is by the W value, which is essentially gives you this kind of closed form way. You have this underlying distribution, and you look at the extent to which your actual real weight, observed weights deviate from that. Or you can actually do this based on setting thresholds. So the real weights are going to be distributed along this data, but they're going to have some sort of bump that corresponds to the real signal. And if you draw if you put a set of threshold and you learn the truncated data, you could essentially also do this kind of procedure and also plot a QQ plot and see to what extent your given weights deviate from that. So presumably your real signal is going to be after training deviate upwards, and which is what happens if you describe. You need to actually run this to many thresholds. The other way of doing this is with just directly plugging into W values, which is a closed solution, and you could compute very quickly. So one of the things we're comparing here is these two different frameworks to allow you to now prune your networks and with all the benefits that come with it in terms of lower storage, better accuracy and performance, and so on. So this is hopefully going to be of use to the general machine learning framework. And just to wrap up, if you've fallen asleep, what's the punchline? So one is entropy is the universal measure of structure and information, and you're able to actually there are ways to construct an entropy that helps you with your problem. Entropy theory and optimal representations can lead to impactful developments across math, computer science, and biology through kind of this three-way framework. And I'm thankful to the beginning of my scientific journey for everyone I've worked with and learned from, and these individuals who I've collaborated with over the years, and to you for hosting me today. So thank you very much. If there's any questions, I have to go back, but otherwise, thank you.
Do you have any example where discovered factor that could you use guide to identify targets for?
Yeah, so that's the direction.
Examples, I guess, makes sense. You show things.
Yeah.
You could relate your finding to ground truths.
Yeah.
Can you use that for?
Yeah, that's the most exciting part. So we're collaborating with experimentalists to do that. So I have a close collaborator I'm working on with to extend this to actually learning disease mechanisms and new biology. So one of the exciting things he found is he made a certain connection in his PhD work in platelet biology between these two genes, SVEP1 and SVEP and PAIR1, and which is a new insight that was published and a very new thing. And he actually found a latent factor that makes that same relationship. So we kind of find these connections. We also have to kind of know what you're looking for. So we are starting to collaborate with experimentalists to actually piece together these kind of biological disease mechanisms or these stories, biological stories that help you with hopefully drug target discovery, and working on extending the methods to multi-layer in a multi-layer way or multimodal way, kind of other sort of data sources. So you get kind of this harmonized model that includes all this information in some sort of optimal representation. So yeah, we're kind of going both on the kind of the computational theoretical and the practical direction to find the things that actually people care about. Yeah.
It's in a cocktail party, number of people are increasing.
Yeah.
Is this like is it like a smooth flow of like I'll be less able and less able and less able to get the information out, or is there like some pattern there? Beyond this?
Yeah. So this is a general ICA course. So ICA has been around. This is not necessarily like God uses ICA in a particular way, but in general, if the underlying signal is non-Gaussian, you'll be able to recover it. So if you're adding in the case of framed in terms of the CLP, right? If your underlying random variables are not distributed in a Gaussian way, you'll be able to separate them out just by kind of moving to the frame where that's the least Gaussian.
Irrespective of.
In theory, I don't know of any theoretical limitations. Maybe I don't know, maybe people here know, but I'm not aware of, in the limit, it'll get more and more Gaussian looking as a whole, the sum of the random variables, right? But fundamentally, the components are going to be non-Gaussian, so you'll be able to recover it. The problem is if the components themselves are non-Gaussian. I should say they could be Gaussian, but if they're different Gaussians, you can still distinguish. So that's maybe an important caveat. I skipped over the simulations, but in the simulations, we actually use a spike and slab model that consisted of two Gaussians with different variances. So if they could be Gaussian and you can still recover them, but they just can't be too close to each other. Their variances can't be too close to each other, because if they are, there's a point where you're not able to distinguish between them. In this case, we had one of them had a sigma of 1 and one had a sigma of 3. Thank you very much.